Bacterial transformation through plasmids, or how to make bacteria glow. So we're going to start off with this little guy, E. coli. E. coli is a model organism because its ease of use, ease of culturing, and the fact that its genome has already been sequenced, and it's one of the most common model organisms. So let's take a look inside. Here's the inside of E. coli. It has flagellum to have it move since it's a motile bacteria. There is a capsule around the outside in blue. In the middle of that, there is a cell wall in orange, and then a plasma membrane on the inside in purple. But what's really important for us today is the ribosomes, bacteria's only organelle, and DNA. And this all is taking place in the cytoplasm of the cell. Bacteria have two different kinds of DNA. So one circular chromosome, except for in the E. coli, that's going to be 4.6 million base pairs, and then plasmids, which are smaller, such as the plasmid we're using today, are about 5,000 base pairs, and plasmids are how bacteria transfer information such as antibiotic resistance to each other. So the ORI is one of the non-coding segments on the plasmid. It means it does not code for a protein. It is the origin of replication, where the scissors will come when it's time to replicate. The beta-lactamase gene encodes for beta-lactamase enzymes for antibiotic resistance against antibiotics such as penicillins. This is important because we want our media that we're going to culture our bacteria to have antibiotics, so we're only growing the bacteria that has been transformed. GFP stands for green fluorescent protein. That's going to be the green glow that we're seeing out of our bacteria, and it originally comes from a jellyfish species, A. victoria. The PBAD is the promoter for the arabinose operon, and the job of an operon is to make sure that this whole plasmid is only going to be transcribed when arabinose is present. The ARA-C codes for a regulatory protein for the operon. As we can see here, in the presence of arabinose molecules, which are a form of sugar, it allows the ribosome to attach and start transcribing the proteins from the plasmid. Now, to get this plasmid into our bacteria, we have to do a process called heat shocking in a hot water bath. That opens pores in the bacterial membrane, allowing our plasmids in the surrounding solution to enter the cell. The bacterial cell will start to transcribe the plasmids and translate them into the proteins, allowing for the final effect of glowing bacteria.